From Microbe TV, this is TWiN, This Week in Neuroscience, Episode 6, recorded on May 14th, 2020. I'm Ori Lieberman, and you're listening to the podcast about the nervous system. Today, you're missing the dulcet tones of Vincent, who is recording another podcast at this time. But with me is from Salt Lake City, Utah, Jason Shepard.
Hey, Ori, how are you?
I'm good. Still in quarantine in my apartment. So,
oh man.
Yeah. From New York City is Andres Bendesky.
Hi, Ori. Hi, Nirao. Hi, Jason.
And we have a special guest today from Stanford University, from the Department of Psychiatry and Behavioral Sciences and the Department of Neurobiology, Nirao Shah.
Hi, Ori. Hi, Andres, Jason.
Hey, Nirao. Thanks for joining us.
Thanks for inviting me to be on the show.
So how are you all doing? I guess we can maybe go through one by one and kind of talk about, is what the lab plans are for reopening, how COVID has been for you guys. So Jason, do you want to start?
Yeah, we're actually phasing people back into the lab. So the state of Utah never really went on a full lockdown, and now they're going from red to orange to yellow, and the campus is reopening somewhat. So we can have a couple of people in the lab, face masks, social distancing, 2 maximum per lab space. So it'll be interesting to see how that goes. But I think everyone's pretty excited to at least start to get back into the lab and at least in a limited fashion. But we'll see. I mean, numbers here have been pretty low, but I'm still worried that this is too early.
What about you, Andres?
Yeah, I think we have something similar here at Columbia, New York. As you know, it's been very heavily affected, so things are going to take longer than in Utah. Likely in a month or so, around June, things will start to ramp up slowly with maybe at 1/3 of capacity, and again with the same face mask, social distancing, some sort of testing for people. But yeah, it's going to be slow and maybe starting in a month from now.
Yeah, and what about you, Nirao?
We are entering what Stanford's calling phase 0.5, which is sort of what Jason just described. We will have 2 people per lab per day with social distancing, face masks, and all the appropriate sort of safety precautions going into campus. For the past 7 or 8 weeks now, we've been in lockdown with personnel only allowed to go in to carry out essential functions. So, for example, to test for and maintain critical equipment like freezers, tissue culture cell lines, and also the animal colony.
I can give the lab that I did my PhD in is in the Psychiatric Institute, and they're slowly phasing in people as well. They're expecting to have kind of 1 to 5 people per lab going in as early as the next couple of weeks, 2 weeks or so. But everyone's very nervous. It seems quite fast.
Yeah. And what's your status? Are you in the clinic, or what are you up to these days?
So we're going back June 29th. I put together the last bit of data from my thesis and submitted to Journal of Neuroscience 6 weeks ago, and we got okay reviews. So I'm going to try to do a couple experiments for that before I go back to clinic if the labs open up. So I set some breeders, so we'll have some mice in a couple weeks, and hopefully it'll coincide with re-entry.
Cool.
It'd be nice in the end, the break could be useful.
Yeah. Congratulations. You have light on the end of your PhD tunnel.
Yes. No, so I defended in the fall. So So I just have a little bit of data left that kind of were random experiments and put it together.
So Ori's an MD PhD, so he's a sucker for punishment anyway.
I am one too. I did my MD PhD sequentially.
Okay. So that's a good segue. So Nirao, tell us a little bit about yourself.
Okay, I grew up in Bombay. Now, called Mumbai. And I went to medical school there and got very interested in the basic mechanisms of health and disease in my first 2 years of medical school. And that became more and more of an obsession as I started doing my clinical rotations. And at that time in India, there weren't very many research opportunities before you went into medical school. As you know, in India, as in some other countries, you go from high school to medical school or any other professional school. So in my clinical internship year, every spare time, every spare minute I had, I would schlep up to a different institute in Mumbai, the Tata Institute of Fundamental Research, where there were active labs doing basic biological research, and I worked with Ubaid Siddiqui, working on, as a technician basically, working on fly gestation, Drosophila gestation. And then, I just fortuitously happened to end up in Ubaid's lab. He had extensive experience working abroad, and he said, Nirao, if you're really interested in research, I would advise you to get a PhD, although technically you don't need a PhD after an NT. He said, I would advise you to get, real basic science training and apply to Europe or the United States or both to get your PhD, and then that would be a good way to get a solid foundation in research. So I did that. I went to Caltech for my PhD, where I worked on stem cells and stem cell differentiation. These are neural crest stem cells, embryonic stem cells. This is way before stem cells were all the rage. And for my PhD project focused on understanding how it is that these neural crest stem cells that form the entire peripheral nervous system give rise to neurons or glia. And we discovered instructive cues in the environment of these migratory crest cells that tell these cells, these stem cells, to become Schwann cells or sympathetic neurons or myofibroblasts at the expense of other fates and the expense of self-renewal as well. So from there, after with my PhD, I said, this is great. I really like developmental neurobiology, but maybe as for my postdoc, I should sort of focus on how it is that these neurons, once fully formed, work in circuits to enact behavioral programs. And that's when I sort of moved to Columbia University, and there I decided that at heart I was a cell molecular developmental neurobiologist, so I wanted to take a genetic approach to understanding neural circuits and behavior. And, although memory and these sort of higher cognitive processes are extremely interesting to all of us, those are not necessarily amenable to traditional genetic analysis in mammals. So I chose to work on mice on a system in which evolution's done the work for me. Evolution has, within a species, generated two sets of individuals, males and females, that naturally exhibit variation in behavior and a variety of behaviors. So I figured if I could identify molecular or circuit differences between males and females, that would be a good entry point into understanding which circuits might be mediating those behaviors. So from there, I developed a reporter system in mice to label in an unbiased way all androgen receptor, testosterone receptor expressing cells, identified dimorphism in the brain, and then from there I went to UC San Francisco for my first faculty position, and then recently I was recruited to Stanford in 2016, and this is where I've been.
So there are fascinating differences, as you told us, between male and female behavior, and you've been studying both the developmental roles of sex hormones and also the activational roles of how these hormones are important later in life to mediate differences in behavior. And can you tell us more about these differences and how your new work in the paper we're discussing fits into that broad scheme of hormones and behavior and their effects during embryonic development and later in adulthood, and more recently even cycling behaviors within adults?
Right. So this, Andres, would you just sort of, let me unpack what you said, right? So I'm sure that everybody's on the same page. You used words such as organizational and activational, which in the field of endocrinology, neuroendocrinology, people have sort of assigned very specific functions to and meanings to. So it turns out that there are at least two phases of hormone exposure that vertebrate animals see. The first is, as Andres alluded to, is the perinatal, embryonic, late embryo fetal or just after birth. And this is usually a male-specific surge of testosterone from the testes that takes the bipotential brain. So the brain, like most organs, including the gonads, to some point in development is bipotential. It could either differentiate along a male pathway or a female pathway. So with this first surge of testosterone, the brain sees these hormones, the bipotential brain sees this hormone, becomes masculinized.
So the female stays the default. Is that what you're?
So the ovaries are quiescent at that time point. So the ovaries are not exposed to hormones at the time at which the bipotential brain is exposed to testosterone. So at some level, you could say that's a default pathway, just like the body plan is female as a default as well. If you don't have SRY, then you get ovaries instead of testes.
Right, and
the default implies that it's a passive thing. But of course, you and I and Andres and everybody here in Ori are aware that default in this sense actually means that a different gene expression program kicks in. That takes the bipotential gonad, it turns it into ovaries, or takes the bipotential brain and sort of leads it down a feminization pathway. So default, yes, but still an active gene expression program.
Right. So an active program, but that program would not be altered unless there's testosterone present, right?
Exactly. Right. So that's the first sort of organizational phase of sex hormone action, and this is the work of many folks across many vertebrate species, and at that point, both the gonads in both sexes then go quiescent until puberty hits. And then you get this sort of second, more permanent onset of sex hormone exposure in both males and females. The early organizational phase is thought to be essentially irreversible because it sets into motion cell death, differentiation, and axon outgrowth of dendritic sort of arborization patterns that are then set, it's thought, irreversibly between males and females. That's organization. The activational phase that hits after puberty or at puberty is more reversible because in females, the sex hormones estrogen and progesterone cycle across the estrus or menstrual cycle, and neurons and other cells in the body respond in that sort of cyclic manner to these hormones. In males, you have sort of an ultradian, sort of fast-cycling sort of secretion or production of testosterone, but essentially testosterone is constant in the circulation with some diurnal and ultradian variations. But if you take away testosterone in males, as in adult males, then they lose male typical behaviors. But if you give testosterone back, you sort of regain those behaviors as it were.
So then
these are sort of reversible.
So just trying to summarize, what's fascinating to me, and you're going to sort of delve into this with your paper, is that these behaviors, I think we all sort of grasp that hormones can, regulate behavior on a sort of acute level. But what you're getting at is that the actual structure of the brain, the circuits in the brain, can be influenced by hormones, and you're going to talk a little bit about that. And that plasticity that plasticity is actually reversible with the phasing, right? To me, that's remarkable. You can set up the system with hormones, and that makes sense. But now you're saying, well, a lot of these changes in the brain can happen very quickly.
That's right. Quickly, of course, is relative, right? Here we are talking about a 5-day estrus cycle in the mouse.
Right.
The humans, it's about a 28-day cycle. The ovulatory cycle is 28 days. And other other species, it's going to be different as well. But that's right. So the brain is sort of, if you will, organized essentially irreversibly, people think, developmentally, and then in the adult, it is acutely sensitive to variations in sex hormone levels. That's right.
So many groups for several decades, including Bruce McEwen and Catherine Woolley and others, have discovered synaptic and changes circuits in the hippocampus and in different parts of the hypothalamus that also change with either with the estrous cycle or when you give a progesterone or estrogen. Can you tell us what you discovered in a nutshell, like you're finding that estrogen affects the projections from one part of the hypothalamus to another only in females and not in males. And I'll let you take it from.
So that's right. So, many folks have shown that sex hormones affect processes, neural processes, right? So, of course, work of Dominique Turan-Alran at Columbia, where you are, Andres, showed many years ago, a few decades ago, that developmentally, if you take hypothalamic explants, then outgrowth from those axonal outgrowth from those explants is sort of dependent on estrogen signaling. And that's developmental because those were sort of fetal, I believe, hypothalamic explants from rodents. And then work from Bruce McEwen's lab, where Catherine Woolley was at the time, showed that in the CA1 dendrites, there's a sort of cyclic change, about a 30% change in the number of spines in CA1. And then other folks have shown, and more recently in the cortex, prefrontal cortex, for example, that again, estrogen acting through different receptors can alter dendritic spine architecture as well across the estrous cycle in rodents. Our work is subtly different, but it's along sort of conceptually very similar. It shows that genetically defined neurons, a tiny collection of neurons in the ventral medial hypothalamus, send projections to a different hypothalamic nucleus, the enteroventral periventricular nucleus, which is a mouthful. So I'm going to abbreviate that to ADP. And these projections, the axonal organization to ADP from VMH-VL neurons cycles across the estrous cycle. We see about a threefold change in the number of presynaptic sort of boutons from these axons onto the ADP across a 5-day cycle. So it's distinct in the sense that we are defining a presynaptic plasticity that is regulated by estrogen signaling, which we showed acts autonomously on these VMH-VL neurons. The other difference, I guess, is, CA1 dendritic spine architecture differences are really well documented, but the function of those changes is hard to define. And it's hard to define those cells, those CA1 cells prospectively, because there are no markers for the cells that are showing dendritic spine changes in response to the cycle. In our case, with the ventromedial hypothalamus, we and others have identified a genetically defined set of cells in the VMH-VL. So that's a tiny, tiny subnucleus within the larger ventromedial hypothalamus that express progesterone receptor and estrogen receptor. And that respond autonomously to estrogen to sort of regulate the plasticity of the projections to the ADP.
So, sorry, I think we should step back a little bit in the global picture here. So, what in terms of your studying? So, this part of the hypothalamus, what does it actually do in terms of controlling behavior?
The ventromedial hypothalamus is an extremely heterogeneous region. It sits at the very sort of ventral base of the brain, essentially abutting the cranium ventrally. And it's been served across all vertebrates and is subtly involved in multiple functions. So, for example, in defense from predators, in appetite satiety control, and also reproductive behaviors, such as female sexual behavior. And then shown by classic studies by Don Pfaff and Jeff Blaustein in the 80s and 90s, and even earlier by Don Pfaff in the 70s. And then it's also been implicated in a larger context by Hess and colleagues, by Menno Crook and colleagues and others, as being important for sort of aggression, territorial aggression in males.
Right.
So then, for us, with more modern molecular tools, the question has been, how are these different functions sort of parcellated, allocated within the image?
Yeah, I mean, for the non-neuroscientists, this is something that probably not a lot of people know about, but that the brain is organized in these very small nuclei. We all know about the cortex, the hippocampus, but there are these clusters of neurons or cells that can control a lot of behavior, and a lot of these behaviors are evolutionary conserved, innate sort of behaviors that are super critical, but trying to figure out how these nuclei work in the brain are tough because they're usually deep in the brain, they're small, and as you say, they sort of can have a variety of functions. And so molecular neuroscience is not being, sort of on the front end of dissecting out what these nuclei are doing, which is what you're doing.
That's exactly right. So the region, the hypothalamus that we work in, is extremely well conserved. It's sort of, if you will, as you said, it performs innate primal behaviors that are conserved across all vertebrates, structurally and functionally as well. So yes, the hypothalamus controls a lot of innate outputs and physiology.
So do you want to?
Go ahead, Andres.
No, if you wanted to tell us more about this behavior you're studying and how this female sexual receptivity changes with the cycling, with the estrous cycling, and how now you're finding about this specific part of the hypothalamus can help explain this cyclicity of the behavior.
Right. So in many animals, invertebrates, in many species of invertebrates, the females are only receptive to mating, will only mate when they're ovulating or just around the window of ovulation. So they'll only mate when they're physiologically fertile. And one can intuit that this means that this is a strategy to maximize reproductive success, because you only want to mate when you know you're going to be fertile. You therefore increase your chance of having progeny. You also sort of decrease exposure to yourself in the wild, so you decrease the risk of predation. And if you can control when you mate, then you might also have evolutionarily evolved mechanisms to enhance the level of progeny if they're born in the right season. So this is one of the commoner mating strategies across vertebrates, and mice and other rodents, definitely many of the rodents sort of follow this pathway where female mice ovulate and they're only mating around the time of ovulation, sort of an estrous period, or the period of heat, if you will. And in the mouse, the estrous cycle, which is akin to the menstrual cycle in humans, the estrous cycle in the mouse is 5 days long. And they're ovulating for about, they're sexually receptive to male attempts to mate for about 18 to 24 hours when they're just about to ovulate. So there's a sort of phasic rhythm to female mouse-female sexual behavior that is sort of locked in with the ovulatory process.
So in the introduction to the paper, and we'll describe the title of the paper in a minute and point people to that, but you describe the evolutionary advantages of this strategy, including reducing predation and kind of as a protective role. And so my impression of mice in the wild is that they're not necessarily interacting with each other very frequently, that they might be kind of in these sparser populations. So in some ways, this might be disadvantageous because you have to not only come to a partner, but you have to come to a partner at the right time. Are there examples of population density, kind of populations of rodents that have adapted depending on the density in the local area of rodents? I don't know if that question makes sense so much.
Right. So I think let me unpack that. So yes, mice in the wild tend to be sort of sparsely distributed, or at least they prefer to be solitary, right? But males will eventually find a female mate with them, and then the female and the male have a litter, and they sit together for that duration. And as soon as the sort of female gives birth, she goes into estrus right away. So you can mate with her again. So it's one way of sort of increasing or maximizing the chance of reproducing, because then you have access to the female. Other animals will ovulate when they mate. So those are called induced ovulators. So rabbits, I think, are thought to be induced ovulators. And it's a different evolutionary strategy to maximize reproductive success. If you do that enough times, then just by the sheer number of mating, you will have enough progeny that eventually survive no matter when you've mated. So you find a partner, you mate, you ovulate, you have progeny, and then a certain fraction of that survives. So those are induced ovulators. So yes, there are other strategies. And there are also sort of animals, and humans are perhaps a good example, where the two have been sort of decoupled, or at least they've been made cryptic. Right. So the menstrual cycle is not necessarily locked in with reproductive or sexual receptivity. But there are sort of the psychological literature suggests that there are sort of variations in attractiveness or sort of women's states in terms of their reproductive behaviors. but there's no locking as we see in the mouse.
And so as you guys mentioned before, that these are very conserved behaviors, but in some ways the comparative kind of function of the same circuits that have been adapted to different strategies is very interesting. And is it understood how the structure and function of these brain structures are different depending on the strategy?
No, that's a great question. We don't know how the structure or function of these structures differs. I do think it would be pretty reasonable to say that the ventricular hypothalamus, the VMHBL, which has a rich collection of estrogen receptor expressing neurons, and is important for sexual behavior, is likely to be conserved across vertebrates, regardless probably of the meetings in the specific mating strategy, whether they're spontaneous ovulators like mice or induced ovulators like rabbits. But how their projections change or how they respond to hormones might be different. We don't understand that yet.
Just as an aside, we just so happens this morning we covered this Nature paper on the fly behavior and how they've worked out the circuit for egg laying. And I mean, it's actually pretty amazing that they've isolated one single neuron that projects from the central nervous system to the ventral cord that can activate a series of behaviors that includes, the abdomen elongating and then laying an egg. And the fact that neuroscience has got to the point where we can actually pinpoint a single neuron in a circuit that's necessary and sufficient for as a complex behavior as copulating and mating and that sort of thing. It's just it amazes me.
I think you're referring to the Barry Dixon paper, perhaps?
Yeah. Mm-hmm. And there they worked out almost the whole circuit that can regulate the egg laying, where in males there's a circuit that, so basically, that series of neurons is not active at all.
Mm-hmm. That's right. Yeah, it's from the sex peptide end right after mating all the way to the CNS and then back down.
Yeah, exactly. So that's
to the ventral nerve cord. That's right. That's a beautiful story.
Yeah.
So we should just let's read the title of the paper that we're kind of referring to here. So we'll, for the rest of the episode, discuss periodic remodeling in a neural circuit governs timing of female sexual behavior. And the first author is Sayaka Inui, and then Nirao is the senior author, and this was published in Cell towards the end of last year. So you can follow along with us if you're listening at home.
So you
this was a wonderful collaboration, I should point out. This was a wonderful collaboration between many folks in my lab at Sayaka. Anyway, it was a postdoc leading the story, and also my colleagues at Stanford, Professor Marcus Levine, who is the president of Stanford University, and also Jun Deng, who is an associate professor in neurosurgery.
Better known for his work in the striatum and basal ganglia, which is how I have heard of him, yes.
Exactly.
Yeah, I was just getting at, you found something very cool, these neurons in this part of the hypothalamus that express both the progesterone and estrogen receptor, and that in females, they respond when they smell a male and when they're displaying sexual behaviors. And I guess you expected these neurons to be active more during estrus than outside of estrus when the females were receptive, but you found that their activity is the same, right? And then this led you, your team down this path of finding that it wasn't the activity of the neurons, but how their projections were changing across the estrus cycle, how they were connecting differently through other parts of the brain. Can you tell us more about the story, how this happened?
That's exactly right, Andres. So we were recording from these neurons, the VMHBL, PVL neurons, we call them in the paper, that express progesterone and estrogen receptors. And we were recording these from these neurons, we were imaging calcium activity in these neurons as a female sort of freely interacted with other animals, a male or a female, or an inanimate object in the cage. And we expected, because these neurons, as a former student of mine, Cindy Yang, had shown in a previous study, these neurons are required for female sexual behavior. If you ablate these neurons, then the female is profoundly unreceptive, regardless of whether or not she's ovulating. So we were expecting that these neurons would be active during the act of mating. And indeed, we found that these neurons were active when the female was mating, and they were more active towards male mice than they were towards female mice. So when females investigate other animals, these neurons would show greater activity when they're investigating male versus a female. And in fact, they were about as active when the female was investigating a female as when they were investigating an inanimate object. But there was a large increase in activity when the female was investigating a male. And all of these studies were done when the female was sort of sexually receptive. And we sort of hypothesized that these neurons, because they're required for mating and are active during mating when she's in estrus or when she's ovulating, would not be active when she's not ovulating and she's unwilling to mate because she's not ovulating, she's sexually receptive. But to our great surprise, these neurons were indistinguishably similarly active towards males, females, and inanimate objects when the female was not ovulating or not sexually receptive. So this was a surprise to us. And we said, well, the approach we had been using to image these neurons was looking at bulk calcium activity, and it's not looking at action potential, which is electrical firing of neurons. So it's not as fine-grained an analysis of neural activity as one might want. So we said, well, maybe there are subtle diminutions in activity in these neurons when the female is not sexually receptive and she's not related. And maybe if we boost the activity of these neurons further using experimental approaches, we might now be able to sort of induce females to be sexually receptive even though she's not ovulating and therefore not fertile. So that was the next thing we tried. We boosted activity of these neurons when the female was not ovulating, either using chemogenetic pharmacological approaches or optogenetics using sort of light-driven elicitation of activation of these neurons, the image we have. But with both approaches, optogenetics or chemogenetics, activation of these neurons when the female was not sexually receptive and not ovulating failed to induce females to become sexually receptive. So then your immediate thought was, well, the projections might be changing, or how did that discovery happen? Right. So, this was a period, as I'm sure all of you are familiar with, a period of intense sort of for Sayaka, for myself, and others in the lab at LabMe. So it was a period of intense discussion and introspection as to what was going on. So Sayaka, to her great credit, tried numerous parameters. When you're doing chemogenetics or optogenetics, you can change so many variables to improve the activation of properties, the activation of those neurons. So she tweaked a number of different parameters with chemogenetics or optogenetics, and in every instance, she had the same result. The females were not receptive.
Nirao, just to interrupt, but just to remind our audience what chemogenetics is and optogenetics. So I think we've discussed on previous podcasts optogenetics, which is just making channels in the cell light activatable, and we can put those channels in specific circuits. But I don't think we've talked about chemogenetics, and that's another way to manipulate cells where we can put an artificial channel in the same circuit, but instead of using light to activate the circuit, we have a drug that we can give that goes into the brain and can only activate those artificial channels, right? And so it's another really neat way of manipulating circuits, but it's also easier to do this in distributed networks of cells because you don't have to pinpoint the light. You can just use the drug to activate any channels that are expressed.
That's right. And also, with DREADS, with the chemogenetic approach, you are slightly altering the membrane properties of the neurons. With activational chemogenetics, you're sort of depolarizing the neurons, or with inactivational silencing chemogenetics, you're hyperpolarizing the neurons. So the thinking is that with chemogenetics, you're doing a more subtle manipulation of neural activity. You're either making the cells more likely to be activated in naturally occurring situations or more likely to be inactivated.
Right.
Whereas with optogenetics, you can come in, you express an optogenetic channel, and you can pulse laser, you can pulse light at a specific wavelength, and you can open those channels and induce activity in those neurons at will and at any temporal pattern that you so desire. So there are advantages to both. One gives you exquisite temporal resolution and specificity with optogenetics. Chemogenetics is a little more subtle, and as you pointed out, it's a little more easy to implement. And you don't need any fancy cables running down on animals, along an animal's body or into its head.
Right. I just think from again, stepping back and seeing how much progress we've made in neuroscience, it is amazing to me that we've got such control over these circuits, it's almost like mind control in a way.
Right. Yeah, these tools have been really powerful in enabling us and all of you and other people in the field, and sort of really understanding what the activity of the neurons means in terms of behavioral output or physiology. Yeah, it's been transformational.
Yeah,
that word is used too loosely, perhaps, often, but here it's true. I mean, these technologies have really transformed the way we do neuroscience in so many different areas.
So by directly activating these cells, you can't drive sexual behavior in non-receptive females. Can you just locally apply estrogen through a cannula, for example, or estrogen and progesterone, and activate into the to the PVL, into the hypothalamus, and drive activity locally?
We could have done that. We didn't do that. So that would be one way of providing estrogen locally to those neurons without inducing the entire circuit to become sexually receptive. But here we are sort of interested in characterizing the function of the neurons themselves in relation to the behavior, not the hormonal responsivity of these neurons. We know the neurons are hormonally responsive. When we came to that in a different way later in the paper, we asked if estrogen signaling is required in these neurons for the plasticity of the projections.
But I was hooked on the story. You guys tried everything and you couldn't get them to become sexually receptive. If they weren't in estrus, then what happened?
Okay, so to follow right. So then, like I said, Sayaka was tweaking all these parameters of chemogenetic and optimal stimulation paradigms. And we knew from the literature, Catherine Woolley's work, for example, and also Joan Alron's work, for example, at Columbia. And we said, well, what if estrogen and progesterone are changing the connectivity of these neurons across the estrous cycle? And that regulates the behavior. It seemed somewhat of an outlandish proposition because, people had sort of not really been able to show the functional relevance of changes in dendritic architecture, for example. But we had a good lead-in for thinking about this. And that's because, as I mentioned in a former paper with a former student in Young, she had shown that these neurons, these PVL neurons in the medulla hypothalamus, send 3 major projections to the brain, 2 of which are shared between males and females, and 1 which appeared essentially unique to females. It was so sexually dimorphic. There's a projection from PVL neurons to this nucleus ADPV that we could easily visualize in female brains. But we could not see these projections, or there were very few projections from PVL to ADPV in the male brain. So we said, at that time, the technology we used was giving us an average signal of the connectivity of PVL to ADPV. So we said, what if we use a more specific marker for presynaptic termini, for axonal termini in these projections, and ask, are these termini changing across the estrous cycle? And we sort of said, well, if the ADPV appears to be dimorphic in this projection, in the seat of projections from the PVL neurons, maybe this is where we're most likely to see plasticity in presynaptic termini. The other reason to think that is the ADPV is sort of been thought to be a reproductive hub in females from multiple different studies, from multiple investigators, not our lab. So this seemed like a reasonable idea to try. And at the time, we had no other sort of out. Right? We could imagine that estrogen and progesterone signaling in these neurons would change gene expression profiles, and that might change the behavior. But we are interested in more functional readouts of those gene expression changes. So we express in PVL neurons genetically a marker, a presynaptic marker, which is basically consists of a fusion between synaptophysin and a fluorescent reporter. In our case, it was M-cherry. It could be any other fluorescent reporter. And then we express the synaptophysin fused M-cherry in these neurons and ask, do we see changes in the number of M-cherry positive puncta in these projection regions across the estrous cycle?
So to put that in perspective for the people who are not so expert in this, it's just like how many synapses this region is making with this other region, right? That's
exactly right. Yeah. So synaptophysin M-cherry provides a good surrogate to look at presumptive synapses in target regions of projections. So we did that, and we saw specifically in the ABPV about a 2.5 to 3-fold increase in the number of presynaptic termini, or boutons, if you will, that in the ABPV emanating from PBL neurons. And at diestrus, which is a period at which females are maximally unreceptive and not ovulating, we saw a minimum in the number of presynaptic termini boutons. And at estrus, around ovulation, when the females receptive and ovulating, we saw a large increase in the number of these presynaptic termini. And we verified that these were bona fide presynaptic termini because we could label these M-Cherry labeled puncta with VGLU2, which is a marker for excitatory neurons. We know from previous studies that PBL neurons are excitatory, they express VGLU2, and VGLU2 localizes to presynaptic termini to boutons. And that's been known. And we saw colocalization of VGLU2, an endogenous marker of presynaptic termini, with our transgenic marker and synaptic infused M-Cherry, telling us that these were most likely bona fide boutons that were changing in number across the estrous cycle.
So when the females are not sexually receptive, they have as few connections between this region of the progesterone neurons, estrogen neurons, and the ABPV as few as males. And when they're sexually receptive, they have 3 times as many. Like that's yeah.
Right. We didn't directly compare the diestrus female to the male, but it seems similar.
Yeah. It's just an amazing change. Like in different parts of the estrous cycles, the brain circuits of a female will look much more similar to a male, and in other parts, when they're behaving more differently, they may look completely different.
Let me circle back to something that we were discussing earlier on. We brought up the idea of organizational versus activational changes in circuits, irreversible versus reversible changes in circuits, and response to sex hormones developmentally versus adult life. So we were interested in, SAC was interested in asking, are the male PBL projections to the ABPV, can we induce female type projections in the males? And there are 2 possibilities, right? One is that adult testosterone in males is suppressing these projections. Or it could be that because males lack enough estrogen and progesterone in the circulation, that if you provide estrogen and progesterone to males, we could induce this plasticity. So we tried both experiments. We castrated males, removed testosterone from circulation, and asked, do we see an increase in projections? And we don't in males. And then we castrated males and gave them estrogen and progesterone to sort of simulate the hormonal state of the female when she's ovulating. And we asked, do we now see an increase in the number of presynaptic termini of PBL neurons in males and to ABPV? And we didn't. Suggesting that this plasticity of projections is hardwired organizationally so that females are responsive to adult changes of estrogen and progesterone, but males are not.
It's very cool. So almost like a 2-hit model that you, and this goes to the setting up of the circuits early, and then having the ability to have this plasticity later in adulthood. I think that's pretty unexpected. Yeah,
that's right. Just as I said, hardwiring of plasticity seems just counterintuitive because that's not the way we typically think about plastic processes in the brain. That's right. And it's not that the male neurons, the analogous neurons of the males, don't express estrogen receptor and progesterone receptor. We and others have previously shown that these neurons also express sex hormone receptors in the males, but they're just not responsive in a similar way as females.
What do you think happens to these projections that are maximal in estrus? Do they retract all the way, or do they get degraded? And then how far do they go back?
Right. We think, we didn't really discuss this in the paper, but we use a different marker. We use membrane GFP to label the processes of PBL neurons into the ABPV as well. And we saw a net decrease in membrane GFP fluorescence as well that cannot be accounted for simply by changes in bouton number. So it seems that there might be some local extension pruning of axonal processes. We don't think they retract back all the way. It's a millimeter away. The VMH, we have the PBL neurons are a millimeter away from the ABPV. And across a 5-day cycle, that would seem to be sort of the Herculean task to retract processes of back a millimeter and then to project them out a millimeter every 5 days. We are hypothesizing that it's local changes in architecture. So that's where we're going next. We are visualize the dynamics of these photons in vivo in females.
So is there any difference between virgin females and experienced females in this plasticity?
That's a great question. We didn't test that. Virgin females will mate so long as they're in that sexually receptive ovulatory phase of the cycle. But there's definitely some, even though the behavior is innate, sexual receptivity, there is some increase in receptivity with experience. So it could be that there might be some subtle differences between virgin females in these projections versus sexually experienced females. We just haven't tested that.
So you find that inactivating these neurons from the progesterone estrogen neurons from this part of the hypothalamus VMH to the AVPV stops the sexual receptivity, but activating the same neurons does not stop does not induce the behavior. But can you elaborate on why you think the difference is? You're not stimulating at the right pattern, or you're not stimulating the release of the proper transmitters, or because these neurons project to 3 different places, you're stimulating things that go in both directions and not just the projections that go to this region? Or what do you think accounts for this difference?
So when we use hilarabsin, which is an optogenetic actuator that hyperpolarizes and therefore silences neurons, when we express hilarabsin in PVL neurons and implant a fiber optic cable specifically over the AVPV termini, even at a phase when the female is receptive and has maximal projections into the AVPV, if we inhibit those projections, the females are sexually receptive. And in fact, we can cycle light the laser on and off in the same assay in the same female when she's sexually receptive. If the laser's off, she will mate with the male when she's sexually receptive, because she's sexually receptive. But as soon as the laser comes on, she runs away, rejects the male, kicks in, and the male can't really succeed in mating with her. So every 5 minutes, we could sort of cycle the same female to be receptive versus not being receptive. And we were using wild-type males, so we had a sort of an orthogonal control, if you will, in the same assay. And the wild-type male's behavior towards this female was the same, right? He would try and mate with her when the laser was off, and he could succeed, and he could even ejaculate. But if the laser was on and she was rejecting him, she simply could not mate successfully to ejaculation. The optogenetic stimulation experiments, or the chemogenic stimulation experiments, you're exactly right. We tried cell body stimulation. We didn't do terminal-specific stimulation. One of the issues we came up against is just plain old biology. When females are sexually receptive, they're already sort of, if you will, maximally sexually receptive. They will lordose, they will adopt sexually receptive posture, and they will accept male advances, they will not run away, they will not kick him. So we had already, if you will, reached a ceiling of fact. So one reason we might not have seen an increase in receptivity when the females are already receptive when they're stimulating these neurons is we are already at the maximal receptivity she can exhibit. On the other hand, when she's not sexually receptive, when we don't have these elaborations of projections in the AVPV and we stimulate these neurons, our data would suggest that in fact there's ineffective communication between PVL neurons and AVPV neurons in the absence of those projections. So that we can't induce downstream activation of specific neurons to elicit the behavior. And one way we showed that is we express, the DREAD activational DREAD actuator, DREAD GQ in PVL neurons. And then in sexually receptive versus sexually unreceptive females, we activated these PVL neurons and asked, do we see activation of postsynaptic cells, putative postsynaptic cells in the AVPV that should express the marker FOS upon activation. And we saw that when the females are not sexually receptive and they're not ovulating and they don't have maximal projections into the AVPV, when we activate these neurons, we see some FOS induction in the AVPV, but we see far more FOS induction in the AVPV when the female is sexually receptive, is ovulating, and has maximal projections, suggesting that there's actually more effective communication between the pre- and the postsynaptic neurons when the female is in estrus. And we sort of confirmed that notion by also doing, and this is the work of Renji Young, a graduate student in the biology program at Stanford, who's in Jinping's lab, with whom we have a fantastic collaboration. And Renji showed that even the spontaneous activity in putative postsynaptic cells in the AVPV differs in females that are receptive versus females that are not receptive. When the females are receptive and have maximal innervation of the ABPB, you see far more spontaneous events in these ABPB neurons compared to when she's not related, not receptive. And similarly, when we did optogenetic circuit mapping, this is sort of a complicated experiment, so you guys might have to unpack it further from what I'm going to say. When we express the optogenetic actuators, channelopsin, which is an activational actuator in PBL neurons, and then have these PBL neuron fibers in the ABPB, we cut a slice across through the ABPB with these fibers from PBL neurons in the same slice. And then we can stimulate these fibers with laser illumination and patch onto single ABPB neurons that are putatively downstream, postsynaptic from PBL neurons. And when the female is not on estrus and not ovulating and has fewer projections, when we stimulate these projections, these termini, with light, and patch on the putative postsynaptic cells, we see a far smaller evoked response compared to when she is in estrus and she has maximal projections. So even though at some level this laser illumination is sort of a sledgehammer approach to activating these termini, we still see about a 2.5 to 3-fold change in the amplitude, the sort of synaptic communication between PBL neurons and the ABPB neurons in the two different phases of the estrous cycle. So suggesting that really, you get better or more effective communication between pre- and postsynaptic cells in the females ovulating.
And this may highlight more generally the role that these sex hormones might be playing beyond these cells, right? That they might be affecting the circuits more broadly in the ABPB and in other parts of the brain that are necessary for this behavior, right?
That's right. So, the ABPB itself also has cells that express estrogen receptor and progesterone receptor. So it could be that the hormones are acting it along every node of the axis.
So, Nirao, what's the most exciting future directions that you're going to move on from here?
Yeah, one of the things that we are really interested in is understanding, as Andres was asking me, what are the dynamics of these presynaptic termini of PBL neurons? Right. So we, a lot of people have been extremely interested in synaptic plasticity for several decades. Here we have a nice platform in which we can prospectively label neurons, the same neurons from animal to animal, and ask what's happening to endogenous presynaptic presynaptic gluton formation versus pruning of those glutons in real time in sort of a physiologically relevant setting. So we're very interested in those dynamics, understanding what changes across the estrous cycle and how is that controlled at the level of PBL neurons? What are the molecular changes that enable this plasticity,
Yeah, no, I think that. I'm of course very fascinated by this as well, and I think you mentioned this earlier that a lot of plasticity in the conventional sense has always been the structure changing of the synapse, but the postsynaptic side, and this is a presynaptic structural change, and for non-neuroscientists. This is interesting because these structures are extremely complicated. Each side of the synapse has hundreds of proteins, and so building the synapse and dismantling the synapse is not trivial. And you're seeing these changes, within a few days. So I think dissecting out the mechanisms of how that's happening is going to be really fascinating.
Yeah. And the other thing this will let us do is ask, what happens to the corresponding postsynaptic side?
Yeah, exactly.
Postsynaptic dendritic plasticity, spine plasticity that these presynaptic termini synapsing onto.
Right.
And that's completely unknown, but that's now technically feasible with the microscopy technologies that people are using.
Right. Very cool.
I have a question more into the realm of speculation, maybe a harder question about what do you think the implications.
A more dangerous question, perhaps.
Yes, the dangerous question to save it to the end. Like, what do you think are the implications for humans, in the sense that many of the cells you describe and the circuits are dependent on pheromones through an organ that humans don't have, right, and that sexual receptivity in humans is mostly decoupled from the from the menstrual cycle. These are fascinating studies. And how do you think they inform about what can be happening in humans, or do you think it's completely different? These cells are not important. Are they doing something else?
Right. So let me answer the specific question first. The ventromedial hypothalamus is conserved in humans, and this is really hard to sort of image with even the latest MRI magnets that people are willing to enter. It's very hard to image in situ in sort of, in volunteers. But clinical studies have shown that this VMH is likely to be playing a role in sort of aggression, maybe appetite control, and also sexual behavior in humans. Although realizing the huge caveat here is these lesions, naturally occurring lesions or tumors in humans tend to be widespread. So it's hard to say, this is exactly the VMH. The VMH is a tiny, tiny structure, even within the hypothalamus. And the VMH-VL neurons that we are studying in a mouse only sort of has about 1,500 to 2,000 neurons. So to image that or to localize the pathological lesion to that is very hard, if not impossible. So let me start back further from that. The more general question you're asking, I think, which is where you want me to go, is how is this generally relevant to sort of, humans in terms of the menstrual cycle and behavior? And there have been a couple of studies, including from Vincent Provost's lab, for example, fMRI studies, describing sort of bold changes in fMRI signal across the menstrual cycle. The functional correlation of that we don't understand yet. But from the disease side, it is really well known. People talk about premenstrual syndrome, and women sort of have reported for many, many years, all these sort of mood and anxiety and appetite changes they encounter as part of the premenstrual syndrome. But less well appreciated, but clinically well documented, is that a large subset of women have migraines or epileptic epileptic episodes that sort of coincide with a specific phase of the menstrual cycle, different phases of the menstrual cycle, in fact. So some women will report epileptic seizures during a specific phase of the menstrual cycle. Other women will report epileptic seizures during a different phase of the menstrual cycle. And a smaller subset of women, the numbers vary, the percentages vary depending on what article you read, but somewhere between 1% and 5% of women suffer have premenstrual dysphoric disorder, which is a DSM-5 sort of diagnostic a condition listed in DSM-5, the psychiatric sort of manual for mental health illnesses. And this manifests sort of across in specific phase of the menstrual cycle as severe changes in sort of mood, anxiety, appetite, and sleep that often require clinical intervention. So there are lots of data in the human literature that suggests that in health as well as in sort of more pathological states, there are changes in behavior across the menstrual cycle. How this is sort of what the mechanistic basis of this is poorly understood, and it's very hard to study, as you can imagine. But our studies suggest one potential mechanism whereby you could have changes across, changes in estrogen and progesterone in circulation, that the brain could be rewiring actively, for example. And we intuit that at least with these PVL neurons, that the brain rewiring that we're seeing is going to be underlaid by changes in gene expression. So then, if you step back further, the estrogen and progesterone, which act on transcription factor type receptors in any event, are going to cause gene expression changes that you can easily imagine lead to changes in neural excitability and function and physiology. So it could be that, this is the way we see manifestations in humans, not necessarily with sexual behavior phase locking to a specific point in the cycle, but more sort of other cognitive level changes in behavior and in disease states, seizures and migraines and other conditions. So there is clinical correlation, not directly with sexual behavior as you surmised, but with other conditions.
Any more questions for Nirao?
No, I
so
think
many, but we would be here all day.
Yeah, I think it's fascinating. I mean, we didn't touch on the evolution of the circuits themselves as well, but I've asked Andreas about this as well, about how do you select for circuits that can mediate complex behaviors like this, and then have them become innate in a way that you can wire them up. Now you've got an extra complication of making them plastic on top of the wiring, and so it's pretty amazing.
Yeah, you work on periscopes, Andres. Let me ask you a question.
You're the guest. Go ahead.
Okay, so may I ask a question though?
Yeah, no, of course.
Are periscopes induced or spontaneous oscillators?
They're both spontaneous. They also cycle like 4 or 5 days, like the lab mice you study.
They're spontaneous.
Very similar. Yeah.
Okay.
Yeah. And this is, I think, Nirao's question is important because we work with a peromyscous species. Some are monogamous and some are promiscuous, and relates to the question Sori was asking initially. Depends on the density, and our monogamous species live together for a whole year, and especially in the reproductive season. But they're still spontaneous ovulators. They don't. They don't vary based on their mating system.
Right.
So wait.
Go ahead.
So no, I was just trying to get. So this one species is monogamous or non-monogamous, depending on the density. Is that what you said?
There are some correlations if you look at cross mammals, like 2,000 species of mammals. The monogamous ones there's only like 8% of mammalian species are monogamous, and they tend on average to live at lower densities than the promiscuous species. But there's not a super strong correlation, but there's some effect.
But different peromyscous species, correct me if I'm wrong, Andres, are monogamous or non-monogamous depending on the specific species.
Right, correct. But those species the monogamous ones tend to live at lower density than the promiscuous ones. Yeah.
Cool.
I actually realized that I have one more question. So is there cortical top-down control of this circuit at all?
Not so far as you can tell in a monosynaptic way.
Okay.
So we don't see direct sort of, for example, prefrontal or cingulate projections to the VMH, but that doesn't mean that there isn't a polysynaptic, neurocircuit that influences these behaviors. And, you asked me a great question. I think it was Cushing, the famous physiologist, maybe 100 years ago, who said, one of the prime functions of the cortex is to inhibit these sort of primal behaviors of hypothalamus. So one would be very interested in figuring out, how does the cortex influence these sort of primitive innate drives, like sexual behavior, for example.
Right. Yeah, and whether it could do it through blocking structural plasticity would be very interesting.
Right. Yeah, we don't think it's going to be a monosynaptic pathway. It could be certainly more circuitous.
Cool. Okay. So we can wrap up. We'll do listener emails on the next episode next month. So you can find us at Apple Podcasts or other podcast players, or at microbe.tv/TWiN. We'd love to get your questions and comments. We'll read them next episode. You can send them to TWiN, TWiN, at microbe.tv. Consider supporting the science shows of Microbe TV at microbe.tv/contribute. And I wanted to thank our guests today. So from Stanford University, Nirao Shah.
Thank you, Ori, Andres, Jason, for having me here. It's been a pleasure. It's been fun. And I'm hoping I'm back again at some point with you guys.
Yeah, thanks, Nirao. It was great to have you on.
Yeah, thank you.
Thank you. It's a pleasure.
From the University of Utah, Jason Shepard. You can find him at Jason Synaptic on Twitter.
Thanks, Ori.
And from Columbia University, Andres Bendesky at Bendesky on Twitter.
Bye.
And you can find me at Ori Lieberman on Twitter. Thanks for listening, and we'll see you next time.